Muscle contraction. So we had talked before about the different smaller subunits of a muscle. The muscle as a whole is an organ. You treat it like an organ. It has its own connective tissue sheath called the epimysium. Deep to that, or the smaller subunits of that whole muscle rather, are fascicles, each with its own perimysium connective tissue. Fascicles are made up of muscle fibers. Muscle fibers have their own endomysium, as well as their own specialized plasma membrane called the sarcolemma. Within the muscle fiber, this is where we're at now, muscle fibers. That's the AKA skeletal muscle cell. We can't call it the skeletal muscle cell. We'll always call it the muscle fiber for exam purposes, but this is just for clarification so that we're all still straight. This long tube-like cylinder-shaped cell is really made up of a bunch of smaller organelles called myofibrils. Myofibrils are unique and complex organelles made up of those protein strings we call myofilaments. Myofilaments are protein strings bundled up together into sarcomeres. Remember we talked about that. A length of myofilaments with their sarcomere longwise divisions. Bundle of those makes up a myofibril. A bundle of myofibrils plus other typical organelles you find in a cell, any old cell, is a muscle fiber. Muscle fibers are long, they're cylindrically shaped, they're multinucleate, and they have tons and tons and tons of mitochondria. So multiple is even an understatement, it's like lots. The reason that they're multinucleate is because in development, like in utero, they were different cells that fused eventually. So now they're long individual cells, but when you were still developing in the womb, they were individual cells. And they retain their mitochondria even after you're born. So these are long multinucleate cells with lots and lots and lots of mitochondria. Notice that the nuclei are all sort of pushed to the outside of the cell. These cells are so jam-packed with myofibrils and mitochondria that other stuff kind of has to get pushed to the outside. That's why when you look at skeletal muscle fibers, you have all your striations here, you find your nuclei out like that. They're on the outside of the cell. Muscle fibers have their own specialized stuff in addition to this unique organelle. They have a modified plasma membrane called a sarcolemma. It's a regular old plasma membrane with some specialized features. And because it has some specialized features that are unique to the cell, it gets its own name, sarcolemma. It also has its own modified cytoplasm, which is rich with glycosomes and myoglobin. Think about the root words of these. So some refers to like organelles or like little vesicle bundles like lysosomes, right? Exosomes. So glyco refers to, what do you figure? Glycogen, sugar chains, those big bunchy sugars you remember back in the chemistry of life lectures. Those simple sugars will arrange themselves into sort of medium-term storage bundles called glycogen. Glycosomes are organelles that hold on to glycogen. Let me actually get rid of this and then say organelles that contain glycogen. Now, myo refers to muscle globin. Is like a protein. right? Like hemoglobin is a complex protein that holds on to iron molecules so that it can carry oxygen. Well, interestingly, this is also it's like a kind of a modified hemoglobin. It's just special to muscles. So this is like hemoglobin. In that, it's a big complex protein that has iron molecules that holds on to oxygen. Now, why would a muscle have these special organelles and have a lot of them, as well as a lot of mitochondria? Energy. Energy, right? Energy. Mitochondria make ATP. ATP is the basic currency for energy in the cell. Glycosomes hold glycogen. Glycogen is fat bundles of sugar. You need sugar to fuel mitochondria to make energy. What else do you need to give mitochondria to make energy? Oxygen. Myoglobin, like hemoglobin, hangs on to a lot of oxygen. Having lots of glycosomes and myoglobin means that the mitochondria always have food, and they always have backup food storage for when there's not anything floating around free in the cell. This helps your muscles contract and maintain your posture continuously, even when a given muscle fiber might not be getting much in the way of food or calories. And again, that sarcolemma, the unique special plasma membrane to a muscle fiber, is in turn surrounded by a connective tissue layer, a very thin, wispy, dense, irregular connective tissue called endomysium. So you'll notice I repeat myself a lot on these slides because I know that these terms get confused so easily. You'll hear me slip up, I'm sure, in these recordings, and I won't even necessarily catch myself where I make a mistake. I say myofibril instead of muscle fiber, something like that. But we really have to harp on it because they are discrete terms and we need to keep them separate in our heads as best as we can. So again, the functional cell of muscle tissue, a special skeletal muscle cell, is called the muscle fiber, and we will always call it a muscle fiber. We will not say skeletal muscle cell. The organelle that is unique to muscle fibers, the major one that does contractile stuff, is the myofibril contractile organelle. And the functional unit of a myofibril is a sarcomere. A sarcomere is made up of those myofilaments arranged in regular repeating patterns and then bundled up. This right here is a good illustration of a myofibril containing, I'm going to use a different color here, all your individual little protein strings bundled up. Okay, protein strings, myofilaments. Okay, but you see how they overlap and it creates this nice regular stripy pattern that makes the striations, right, on a skeletal muscle cell and a cardiac muscle cell. So a length of that, a vertical length of that, is called a sarcomere. This is the basic functional unit of a myofibril. It is the smallest contractile unit of a muscle. Each length of those will contract, but individual muscle fibers don't get shorter. Okay, or sorry, see there, I made that mistake. Individual myofilaments themselves do not contract. They slide past one another to pull the length of the muscle fiber shorter, to contract it. But the smallest unit that actually does any wholesale contraction is the sarcomere. You find sarcomeres only in striated muscle, skeletal, and cardiac. So that leaves what that must not be striated anymore? Smooth muscle and smooth. No. Each myofibril is made up of a bunch of those protein strings, those myofilaments, made up of myosin or actin or titin. Okay? Myosin is a type of protein string. It's a muscle filament, a myofilament, and that we also refer to as the thick filaments because they are just sort of thick and beefy relative to other stuff. Actin, which if you recall is the cytoskeletal protein that helps keep cells, like the plasma membrane, keep its shape. It supports microvilli, things like that. They're very thin and wispy individually. We call them thin filaments. Finally, we have titin, which are very, very elastic. And these are going to kind of help sarcomeres hang on to other sarcomeres. It also helps rebound so that when you contract, you can go back to the same length again. And when you're stretched, remember muscles do not stretch themselves, they are stretched by other muscles. When a muscle is stretched, the muscle can go back to its regular shape. During contraction, your muscle filaments, your thick and thin filaments, myosin and actin, slide past each other, and nothing shortens except for the unit as a whole. So this protein string here, this actin filament, this thin filament, thick filament, each one of these will stay the same length, but the thick and thin filaments will slide past each other to overall shorten the length of the sarcomere. Okay? Let me erase all of this. So let's talk about the arrangement of those fibers in the sarcomere and how they're oriented against one another during the process of contraction. Now you might be saying to yourself, why are we talking about the process of contraction in this kind of detail with all the stuff in it? I don't know what contraction is yet, really, like on a molecular level, so how can I understand these relationships? I'm going to talk very basically about where the fibers or the filaments move next to each other so that when I go to talk about the actual movement of things attached to the filaments, you'll understand what's nearby and what's available to participate. You're also going to see a video, like I have a video embedded in a few slides that will illustrate this really, really, really beautifully at a molecular level. You're going to need to understand all these steps, but with these notes and with that short little animation, you should be able to put together the whole picture of contraction. So the sarcomere is the most basic contractile unit of the cell. The fibers in the sarcomere are staggered, right? Here's one, and then here's one. Here's one, and then here's one, right? Boom, 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 boom. They're staggered against one another. And since they're staggered, you have different places where things overlap differently, okay? So first things first, I'm actually going to work at the bottom of this first list and work my way up. So the M line, you can think M for middle. Middle of myosin. At the middle of myosin. This is made up of a protein called dystrophin, and it anchors the sarcomere as a whole. So during contraction, you can see this top picture is relaxed and the bottom is contracted. Here's the ends. The M line stays in the same place, but the ends move closer in towards it. The M line at the middle of myosin features dystrophin and anchors the sarcomere relative to the ends of itself and within the cytoplasm as a whole. So these are actually attached to the circle or to the outside of the myofibril, and it's anchored fully so that when the ends of these actually contract, they all pull on one another, and there's full contraction that way. Remember, with your body, you really need, for contraction to do any kind of work, you really need it to be attached to a rigid structure, right? If you just had muscle attached to a glob of fat and you contracted that muscle, you're gonna like see a dent form in your body, or a little bit of a movement, but it's going to be sloppy and uncoordinated and not super robust. But when you attach that muscle to bone, something rigid, you can pull a whole structure much more effectively and get a lot of oomph out of that contraction. You can get a lot of work done with that contraction. So similarly to the action of a muscle as a whole, you have an anchoring, a rigid anchor, like a bone sort of, in the middle of it, that everything is going to contract relative to. The H zone is the space around the M line where you only see thick fibers, thick filaments made of myosin. And it's actually the tails of them. So if you see how each one of these myosin fibers have this sort of like splay shape to them, like that, they sort of have these little like tails. Those ultimately attach to that M line of dystrophin in the middle, and you only really have myosin tails in this area. They're fairly thin, but they're thicker still than actin. Okay? So the H zone, you only see thick myosin filaments. The next region, the A band, features both thick and thin filaments. You have a band of actin, and then you have myosin, and then actin and myosin, actin, myosin, actin. They alternate and they overlap visually. And the I bands towards the ends, you only really see actin filaments. Now you do have those elastic titin proteins. Those are going to anchor the myosin fibers, the myosin filaments rather, to the ends of the sarcomere. But they're so thin and wispy that visually speaking, you don't really see them. They don't contribute to the darkness of that band of the in the striations. So really what you see there is actin. When you see a picture of a muscle fiber and you see the striations, this would be light, a light part, and this would be dark. And this would be light, and this would be dark. The A bands have lots of stuff. They pull up a lot of dye, so they look dark under a microscope. The H zone only has those little myosin tails. That's not a lot of stuff to hold on to dye, so it looks lighter. Pretty cool, huh? It always amuses me when stuff is really just that straightforward. The ends of your sarcomere are called the Z discs. Are called your Z discs. During contraction, you're going to pull the Z discs in towards the M line. M line. Okay? Now remember that the sarcomere is the most basic contractile unit of the cell. So these individual filaments, your actin, your myosin, your titin, your dystrophin, these do not change size. During contraction, they slide past one another, or more particularly, the actin gets pulled in closer to that M line relative to the myosin, the heavy band of myosin. So the overall structure gets shorter, the individual filaments stay the same. That means that the sizes of your bands and the H zone are going to change during contraction. So let me get rid of some of this extra stuff. Okay, just to make things cleaner for this illustration. There we go. Okay, so your I band, which is the space between the end of your myosin filament and the Z disc, which contains only actin, will get shorter. Your H zone, that space in the middle that only has myosin tails and that line of dystrophin at the M line, is also going to shorten. The M line is an anchor, so it does not move. The Z discs come in closer to one another. And the A band, because it's the area of overlap, and the I band and the H zone are shortening around it, actually doesn't change. It stays the same size. This and the M line are the only things that don't really change. The H zone gets smaller, or the H zone gets shorter, the I band gets shorter, the Z discs come closer to one another. The M line, made of dystrophin, it's an anchoring middle line, stays the same because it's an anchor, so by definition it can't move. And the A band, that area of overlap, does not change. The illustration doesn't show that super clearly, but it really just sort of shifts over like that. Now let's look at those myofilaments individually. So we see how they're arranged next to each other, but what are they made of? So your thick filaments, as we've mentioned before, are made up of myosin. These feature 2 heavy and 4 light polypeptide chains that are intertwined. I don't need you to know what they're made of or anything too specific with it, except for what I give you specifically here. Your heavy chain, includes this double helix tail and the heads of the myosin filament. The light chains are sort of like the neck, okay? Your heavy chains are called such because they're bound very, very tightly. They have a lot of mass to them. Your light chains because they don't have as much mass to them. The heavy chains are structural and functional. They give them their bulk and their mass. And functional, these heads are going to be doing the major contractile stuff in the sarcomere. So structural and functional. Okay? The light chains are regulatory, okay? It also has sort of a flexible hinge function to it, and we'll see how that comes into play soon. So now let's look at our actin filaments, our thin filaments. These are made up of globular actin, purple blobs here, surrounded by tropomyosin spirals. Okay? The tropomyosin spirals have their own little extra complex on it that does stuff too, but we're not going to worry about that just yet. Think way back to our basic cell structure or chemistry slides, and we have globular proteins and fibrous proteins. Fibrous proteins generally made structural stuff, right? And globular was doing stuff, like enzymes, for example. It's sort of interesting then to think that this actin filament, which you think of as being sort of a structure, like like a structural component, it's a long string, is actually made up of globular proteins. But as it turns out, these proteins are all going to do stuff, and they're going to interact with that myosin myofilament. Okay? On the globular actin, you have binding sites. These binding sites are going to bond with the myosin heads. So you remember you have that tail, and then a couple of light chains, and then a myosin head. They're going to interact with an active site on the actin. Indeed, on the myosin heads, there is an active binding site. Okay? On actin, they're globular proteins. They have a myosin binding site. They're active sites. This type of protein is called G-actin. They're made out of what's called G subunits, and they're polymerized into these filaments. You don't really need to remember that so much, as they're long strings of functional proteins. So back to the spirally stuff. Tropomyosin spirals around your actin filament. This does two things. It stabilizes the molecule, kind of just helps keep it all, held together. And it also has regulatory activity. These tropomyosin fibers also have studs on them called troponin. Troponin are also globular proteins. They're anchored onto tropomyosin, which is a long strand of protein. It's the spirally protein. Tropomyosin spirals. This is the best mnemonic I can come up with. There's not many that are good. Not many that are good here. And troponin is in the spiral. Okay? These stud the filament periodically. Okay, so you see these every so often. The purpose of the troponin is to act like a switch to move the tropomyosin. So the tropomyosin spirals around, but isn't necessarily static. It can shift around relative to the actin if it wants to. So you see how it's drawn covering up those actin binding sites? The troponin complex can bind to calcium that's released during excitation contraction coupling. That's a whole other set of slides. It's going to bind to calcium and move the tropomyosin out of the way of those actin binding sites so that they're exposed. And when you're not doing contraction related stuff, they're going to be covered up so that you don't have contraction when you don't mean to have contraction. Okay, so don't get scared. I know this slide looks like a lot. It is a lot. But the next slide is it links to anyway, an animation of all this happening. So what I really strongly recommend you do, take this slide, maybe print out a separate copy or have a separate copy handy, watch the video, and reconsult this slide as you watch the video. Pause it occasionally, go back to old slides to remind yourself of what filament does what and is where, but have these handy and then just watch that over and over. I kind of hate to be like, okay, just go to YouTube, but really this is sort of the best visualization that I've ever seen of this process happening. So I strongly recommend that you take advantage. Again, the functional unit of contraction, the most basic unit of contraction, is the sarcomere. Nothing smaller than the sarcomere actually contracts. And contraction of the sarcomere, contraction of lots of sarcomeres among lots of myofibrils, among lots of muscle fibers, is what creates a whole muscle contraction. So remember, you had this actin, double helixy looking chain, and you have myosin, which has those heads. Actin has binding sites. I don't remember which strand is which, but pretend they're on the same strand. And myosin has binding sites too. In addition, you have a tropomyosin, right, that covers all those actin binding sites, and it has its own troponin heads attached to itself. Okay? So the first thing that's going to happen, you have this zap from a nerve that's going to send a signal down and release a bunch of calcium into the muscle fiber. Details coming later. Calcium comes in and binds to troponin. The troponin will then change shape and push that tropomyosin out of the way of the binding sites. Now the actin's binding sites are shown, they're visible, they're accessible by other stuff in the cell. Calcium comes in and attaches to troponin. Troponin acts like a light switch, flips itself a different direction, it changes shape and pushes tropomyosin out of the way. For this part, as long as you have calcium coming in, whenever that tropomyosin comes back, you can always keep flipping it out of the way and keep those actin binding sites exposed. So the amount of calcium in the cell is a rate limiting step. If you don't have enough calcium, you can't keep those actin binding sites visible or accessible. So it doesn't matter what else is happening in the cell. If you don't have enough calcium, you can't have contraction. Your binding sites have to be exposed for binding stuff to happen, and contraction, as it turns out, is a big binding situation. So now your actin binding sites are exposed. You have calcium coming in that's keeping those visible. What's happening on the myosin side, let me actually connect these two. So this down here basically says the same thing I wrote up top. The amount of calcium you have in the cell is rate limiting. The myosin stuff is all in these boxes below, and we're going to start over here. for the moment. It's just, frankly, it's just convenient. So ATP is going to bind to one of these heads. And it's going to cock it back to activate it. Like if you cock the hammer of a gun, I suppose, so that the gun can fire, same sort of thing. ATP binds to a myosin head, and then it cocks back into a ready position. That activated myosin head is going to help ATP engage in hydrolysis. The P is going to pop off, and when it does that, it'll go away, and the myosin head will bind to a nearby actin binding site at their respective binding sites. As it does this, it's going to pull the whole thing over. So really, this head is going to be more like here with its binding site, and it's going to pull this whole thing over. So instead of that actin being here, it's going to get pulled and now be more like here, closer to the M line. The pivoting of that myosin head that is now bound to an actin binding site after ATP hydrolysis pulls the thin filament towards the M line for contraction. ATP will then come in again because you have this constant influx of ATP. It'll bind to the myosin head. Remember when ATP bound to the myosin head, it pulled it back into a cocked position, ready to go. That means it's also going to help it pop off that actin binding site. So ATP comes in, cocks the myosin in a ready position. ATP pops off, myosin binds. Myosin pulls forward and pulls the whole actin chain closer to the midline. More ATP comes in, releases, and pulls it back to a starting position. Now, that sounds like that actin filament ought to just be going back and forth and back and forth and back and forth. But remember, every myosin has two heads, so this is happening on both of their heads all the time. If you take your two fingers, and you know how you can walk your fingers across a desk or something, like they're legs of a person, do that to something that moves, and you can see how you can move, like if you put a piece of paper under those fingers and you move those fingers like they're walking, you can move the paper away, right, in one direction. When you let go of one finger, the other finger's still touching it and still pulling it in that same direction. And in the exact same way, only with binding instead of just touching, your myosin heads are walking that actin filament closer to the midline. And also remember that you don't just have one strand of this. You've got lots. That's sloppy. At any given time. So this is happening along the whole length of your actin and myosin fibers. So your myosin is bound to the M line, which is dystrophin, and walks actin closer to the M line. Actin is bound to the Z disc and is walked closer to the M line. Myosin does not move, it is anchored. The actin is moved closer to the M line by those myosin heads. They walk along the actin heads and pull them closer and closer and closer in. So this is the video I'd like you to watch. I don't think it'll let me play directly in this presentation, but if you click on that link, you'll be able to see it in the PDF. Watch that video, reference the slides, watch it again, reference the slides. I need you to be able to tell me how these filaments move respective to one another, how the different bands and zones may or may not change relative to one another, and the lines may or may not change relative to one another. What's going on when calcium binds, and what does it bind to? What does that reveal? What does that move on the actin? And then what happens when ATP comes and goes on the myosin? If you understand that really well, you'll then also be able to tell me, well, what happens if I don't have any calcium? Can you still have contraction? Why or why not? What happens if I don't have any ATP? What happens? How do you know? Okay. So transitioning away from skeletal muscle for a moment, remember there's two kinds of striated muscle. Skeletal and cardiac. Is smooth muscle striated? No. Smooth muscle is different. Smooth muscle is more elastic than striated muscle. It has a thinner connective tissue covering. And importantly, its attachment points don't line up in those regular repeating patterns that cause a strapy appearance when you look at it under a microscope. Smooth muscles, filaments, are arranged in sort of a diagonal catty corner kind of a way. They also do not have tropomyosin. Instead, they have an analogous protein called calmodulin. They have fewer thick filaments, but the filaments they have more heads. Actins will bind, because they cross-hatch here, they'll bind to one another by these sort of nodes called dense bodies, and that keeps all the different sarcomeres, well, they're not really sarcomeres, but all the different myofilaments organized the same, so that contraction doesn't shift anything inappropriately. And when they contract, because they contract in these opposing diagonal ways, it ends up creating a corkscrew kind of effect. Or like if you take a towel and you wring it. When you wring that towel, it gets a little shorter, right? And it gets a little twisted up and a little bunchier. The same sort of thing with smooth muscle. When those myofilaments get shorter, that's actually the wrong direction, but they're oriented that way. When those get shorter, they tighten up and bunch in the remaining tissue. It gets shorter, it gets bunchier. You don't really need to know that much more detail about smooth muscle. It really functions basically the same way, but things don't line up. You have a couple of different proteins, and instead of you having this smooth, clean, linear kind of a contraction, you have this corkscrew contraction that causes it to be shorter but a little bunchier sort of in appearance. Contraction of smooth muscle, this is the last slide, can be induced externally or internally. This is different from skeletal muscle cells. Skeletal muscle cells rely on the nervous system to stimulate it for contraction. They do not move truly independently of anything else in the body. Your cardiac cells don't have their own inherent contraction, but there are pacemaker bodies in a heart with their own special electrical system that happens and forms spontaneously within the heart tissue itself. The heart does not require the nervous system to keep beating. The nervous system can influence the rate and strength of heartbeat, but the nervous system does not make the heartbeat. That's intrinsic. In fact, if you take a bunch of heart cells and like heart stem cells and you put them in a petri dish, grow them in a vat, whatever, after they've divided however many times and there's a discrete tissue, they will form their own pacemaking and they will start to contract as a whole unit. Pretty cool stuff. For smooth muscle, contraction could happen by internal means. Some some regions of smooth muscle have their own pacemaker cells to create their own rhythmic contractions, or they can be stimulated externally. Now there's more factors involved in stimulating smooth muscle to contract than there are skeletal muscle. Skeletal muscle is kind of nervous system, end of story. Smooth muscle can be induced hormonally, for example, too. So that's more complicated, but just know it can do both. Smooth muscle, especially through the digestive tract, has a contractile pattern that's unique to it called peristalsis. Unique to it in the body, I should say. This is not unique in nature. As you're thinking about these steps, think of a worm, okay? You watch a worm crawling across the pavement or slithering or whatever it is you want to call worms do. And what it does, right, is you see it pinches a little bit and then it shortens up behind that and then it relaxes. And then it pinches a little bit and the tail shortens up behind it and then it relaxes. And as it shortens up towards the head each time, it moves a little further down. Go find a video of it. You'll see what I mean if you can't picture it right now. Your esophagus, your small intestine work the same way. The first third of your esophagus or so is actually skeletal muscle. That's the part you can control for swallowing, but the rest of it's smooth. And the smooth part does peristalsis. Instead of the whole tube moving, though, what it's going to do is it's going to use that same motion to move something hollow within it. That's a bad color for this slide. There we go. So just like I was describing the worm, what's going to happen is you're going to swallow food, which used to be here. More like here is a better picture. In response, your esophagus is going to shrink up roundwise. It's going to constrict roundwise. Smooth muscle always has at least 2 layers in the digestive tract at least. You have a circular layer, so this is your tube at an angle. You have a circular layer that runs like this, and that's the middle, the deepest layer. And then you also have a longitudinal layer, superficial to it, that runs like this. So the circular layer constricts, and when it constricts, it pushes on the bolus of food. Bolus is a lump in the digestive system, a lump of food. Now, if it just plain old squeezed on that bolus of food, you'd have some that squidged out up here and some that would squidge out down this way, right? But you don't want it to squidge back up from whence it came. You want it to go down in one direction. So to do that, the longitudinal layer will shrink up towards the constricted region at the same time. Then when they relax, and the longitudinal layer sort of relaxes back into this direction, it can kind of drag that bolus of food down with it. And since the circular layer is not constricted down here, the bolus can also move in that direction. It's the path of least resistance. So that'll happen again. You have constriction. Bolus could go this way or it could go this way. But this side will shorten up. The bolus will be forced to move this way. And then relaxation will really pull it down distally into the stomach. Same thing with the small intestine, except there it's going to come from the stomach and to the large intestine, which also engages in a very, very, very similar type of contraction, but it has its own extra stuff going on. Stomach, same thing, but it has some extra stuff going on. We're not going to worry about those just yet. The goal of peristalsis in the body is to expel the contents of a tube, so to get that food through your digestive system, or similarly, make sure that urine travels in a unidirectional way, one direction, away from the kidneys and towards the bladder, and then away from the bladder and out of the body and not back up. Smooth muscle, compared to skeletal or cardiac muscle, is capable of producing fewer contractions. It's kind of a slower kind of a contraction, slower kind of a stimulus. But it can maintain tension for much, much longer. And this is important. You don't want your smooth muscle to just twitch, because then stuff can regurgitate back pretty fast. You want it to hold closed so that whatever's going through it can really be forced in one direction and doesn't get a chance to backtrack. So fewer contractions, but they hold their tension for much longer. Not necessarily stronger, but more durable.